রেকর্ডিং এর দিকে নজর থাকবে, কারণ এটা তোমরা পরে পড়বে, এখন না। আজকে কি ইন্টারনেটটা স্লো মনে হচ্ছে? তোমরা কথা শুনতে পাচ্ছ আমার? ঠিক আছে। আমার পিকচারটা একটু মনে হচ্ছে চলে যাচ্ছে আবার আসছে, চলে যাচ্ছে আবার আসছে, মনে হচ্ছে ইন্টারনেটটা একটু স্লো আর কি। এনিওয়ে, the materials, the inorganic materials that are used as nanobiomaterials are mainly noble metals. Noble metal means your gold, silver, iron, they are mainly used. Why they are mainly used? Their main property used as nanobiomaterial is that they are biocompatible. They do not do any harm inside our body. This is the first property. And the second property is that they are detectable by means of the electron microscopes. Some substances are not detectable by electron microscope. Generally, to detect a material by electron microscope, we generally give a coating of gold on that substance. To detect a substance by electron microscope, we generally give a coating of gold, a very thin coating of gold on that substance, because gold is very much detectable by the electron microscopy. So if we use gold or silver as a nanobiomaterial, they becomes very much detectable by the electron microscopy. If we cannot detect anything by electron microscopy, then we cannot know how they functions in our body. To know the mechanism, how they functions in our body, we have to detect them. We have to detect them where it is going and how it is interacting with your protein or your body. So these substances, they are very much detectable, and as well as they are biocompatible. So these are the main advantages. These are the main advantages of using noble metal as inorganic materials. We can use these noble metals as different shapes. As different shapes, you see, this is a nano shell. If we use gold nanoparticle, say for example, we are using gold nanoparticle, we can use the gold nanoparticle as a nano shell. We can use a gold nanoparticle as nano case. We can use as nanostar, nanocluster, nanorod, nanosphere. These all are different shapes of your nanoparticles. And this you see here, this gold material can attach with different biomolecules. These gold molecules can attach with different biomolecules to form a gold biomolecule, nanobiomolecule rather, gold nanobiomolecule. So they can be differently shaped. They can be attached to different biomolecules. They are detectable and they are biocompatible. So we use that gold or silver nanoparticle mainly as nanomaterial, or iron nanoparticle is used as magnetic material. Now, if you see here, if you see here, this is a silver nanoparticle. You see, this is a silver nanoparticle. What it does, how it works, you see here, this is a silver nanoparticle, and this is an antibiotic. Say, for example, this is amoxicillin or ampicillin. They are antibiotics. So this silver nanomaterial attached with that antibiotic. You see here, this silver material is attached with that antibiotic. Now, this is the nanobiomaterial. It becomes a nanobiomaterial. Now, this is a bacteria. Say, for example, a typhoid bacteria enters in our body. Say this is a typhoid bacteria, and these are the DNAs of typhoid bacteria. Now, this silver nanoparticle enters into the inside the bacteria and makes some changes in the DNA of the typhoid bacteria, so that this typhoid bacteria can die. This typhoid bacteria will die after changing the conformation of DNA inside the typhoid bacteria. So if you use nanoparticle, nanobiomaterial, it enters the cell of a bacteria. It enters the cell of a bacterium. You know, the bacterium, a bacteria has only one cell. A bacteria has only one cell. And this nanomaterial, this nanobiomaterials enters the inside the bacterium. Enter inside the bacterium. The, the antibiotic, the antibiotic attached with this nanoparticle, what it does, it breaks the cell wall of the bacteria. Here you see, it breaks the cell wall of the bacteria. These are the flagellus of bacteria. It breaks the cell wall of the bacteria and it enters into the bacteria. And it makes some changes in DNA. It makes some changes in DNA, and due to this changing, the bacteria dies. So this is this is a simple schematic diagram of your how a nanobiomaterial works. Now, there are some other substances I told you in the previous lecture. There are metallic semiconductors. We'll see it later. There are metallic semiconductors as for example, cadmium selenide, or say for example, zinc sulfide or something, zinc selenide. They are metallic semiconductors. They can also used as nanobiomaterial. How it is used as nanobiomaterial, I'm telling you later. They also have your detecting property. We can detect them easily. You see, size tunable emission of quantum dots. That means it emits by UV or IR region of wave, we can detect. And the detection spectra is very narrow. How it is, how they are doing it, I'm telling you just afterwards, with a very short description. Now, the inorganic materials, there are magnetic nanoparticles. You know what is magnetic nanoparticles and how to use them. The iron nanoparticles, you push iron nanoparticle inside your body, and a magnet brings that iron nanoparticle to the targeting area, to take images and to do various things. What are the various things? Proteomics, molecular cell biology, medical science, analytical biochemistry, clinical diagnosis, microbiology, and all these things. The magnetic nanoparticle can be used in all these areas. So these are the nanoparticles. What are the nanoparticles? We saw that noble metals. Noble metal means uh, silver and gold. Mainly gold is used, silver and gold. Some quantum dots, that means inorganic metallic semiconductors. They are also used as your nanobiomaterial, and magnetic nanoparticles like iron oxide. They are also used as inorganic materials as nanobiomaterial. Now, the next section is nanobiotechnology. What is the nanobiotechnology? Integration of nano-size or nanostructured materials, nanoscale analytical tools, and nano devices into biological sciences for development of new biomaterials and analytical toolkits. See, what is integration of nanoside or nanostructured materials? Using of nanosize or nanostructured materials or nanoscale analytical tools to do an analysis by a nanomaterial, or nano devices. What are the nano devices? We'll see it later. Into biological sciences for development of new biomaterial and analytical toolkit, as well as understanding life science. If you use nanomaterial in biological science, the technology to use this nanomaterial in biological science is the nanobiotechnology. Now, the typical characteristics of biological events, that the characteristics of this nanobiomaterial is that they are self-assembled, they assembled by themselves, they are highly efficient and very much specific. It will do where it will go there, where it should go. We'll see it later. Now, we have nano devices. We can use, say, for example, nano robots. They are nano devices to deliver your medicine. Then there are nanopores, there are dendrimers, there are nanotubes, there are quantum dots, there are nanoshells. They all are used. They all are used mainly to deliver medicine in a specific region. That means it is called targeted delivery in a very specific region, or to diagnose a problem in your body. What are these? These are nano devices, nanopores, dendrimers, nanotubes, quantum dots, nanoshells. You see, all of them are from 1 to 100 nanometer. Their length is 1 to 100 nanometer. You see, a white blood cell is of 10 to the power 4 nanometer. So these instruments can enter into the white blood cell easily, enter into the white blood cell easily, and it can target a specific thing of a cell. Say, for example, you have a problem on the nucleus of the cell. This nano device enter into the cell, goes to the nucleus, and deliver the medicine on the nucleus only. This is highly specific. It can enter into the cell and deliver the medicine on the nucleus only, and all other things remain unaffected. So this is a affected cell. Say, for example, this is a affected cell. Normal medicines, what this will do, this is the affected cell, and normal medicine will treat the whole cell. It may damage something here, it may damage something here, but the nano devices only treat the nucleus. That is, this is that much specific. So applications are the applications of nanobiotechnology are to develop of tools and methods, because they are more sensitive, they are more specific, multiplexed. Multiplexed means they can do more than one work, and they are economic because because of nano size, they are very much economic. They can be implemented in the diagnosis and treatment of diseases. They can be implemented in the drug development. They can be implemented in various levels of your medical sciences. And the examples of biotechnology are nanobio devices, nanobiosensors, imaging of nanoparticles, analysis of a single molecule or single cell. If you want to analyze a single cell, you can do it by a nanobiomolecule or a nanobio, or by means of nanobiotechnology. So we first see the nano device. What is the nano device? The nano device, say for example, a nano device is a nanomaterial that is of, say for example, a very nano size. These nano devices are small enough to enter the cell. I told you earlier that it will go to the cell and treat the specific portion of the cell. Say, for example, this thing is disturbing, and this will go here and treat only this thing, not the excluding the and all other things. So you see here, if you want to detect a tumor, say for example, if you want to detect a tumor, then our normal process to detect a tumor, it will get a sign here, it will get a sign here, it will get a sign here, it will get a sign here. But uh, that by imaging, that means by MRI or by something, or by CT scan, you can detect very specifically. But you see, the detection size is somehow larger. But if you use nanobiotechnology, you will see the exact location and exact size. The size of tumor is like it. Now, if you use normal physical exam, blood test or something, the normal physical exam or X-ray, they will show you like that. Here maybe, here one maybe, and here is the good possibility to have a tumor. You have a good possibility to have a tumor here. You are seeing this by means of, say, for example, blood test or different urine test or different physical exams. But when you do image by, say, for example, by MRI or by CT scan, then you will find more specifically it is the tumor is here. But if you use nanobiotechnology, you see the size, you'll find the exact size and location of that tumor. So this is the use of nanobiotechnology, and you see here, nano devices can improve sensitivity. You see, nano devices are very much specific. Now, say for a person, these are normal cells. For a person, these are normal cells. And say this yellow and these are the precancerous cell. Precancerous cell means the person, cancer will develop on that person after some time. Say, for example, after 2 years, the person cancer will develop on that person. Then this cell is turning to this side. This is called precancerous cell. Now, this nano devices enters into the cell and can tell you whether it is precancerous cell or it is a normal cell. So it can, a nano device can enter into the cell and it will tell you whether it is precancerous or a normal cell. And then you can take the precautions on that precancerous cell. How you can take precaution, I'll tell you now. You can take the precaution of this cancerous cell, precancerous cell. You can destroy the precancerous cell, or you can treat the precancerous cell by knowing that the nano by knowing the result of that nano device. Now, you see how we can use the different substances as in the nano device. First one is semiconductor nanocrystal or quantum dots. What is the quantum dot? Actually, I told you previously that the metallic semiconductor, we can use the metallic semiconductors as nanobiomaterial. So what is the metallic semiconductor? you can synthesize metallic semiconductor by cadmium oxide with selenium. You will get cadmium selenide. It is a nano dot, quantum dot, and you can give a coating of zinc sulfide on this cadmium selenide. Now, what this will do? This cadmium selenide nano quantum dot, what this will do? This quantum dot, QT means quantum dot, if you if infrared light or some UV light falls on this quantum dot, it will emit light. If infrared light or some UV light falls on this quantum dot, it depends on which quantum dot you are using, it emits light. When it emits light, you can image that section. When it emits light, you can detect the light and you can image that section. See here, here is the quantum dot. It is inside the body. Here is the quantum dot, and if you if you give infrared radiation on that quantum dot here, it emits light and it is detected. It can be imaged inside the body. So what is the requirement of this? You see here, this is a rat. Say for example, this is a rat, and this rat has a tumor here, or this rat has a tumor here. You have injected quantum dot to this rat, and the quantum dots will be accumulated on that tumor only, and the quantum dots will go and will be accumulated on that tumor only, and nowhere in the body of the rat. If you have tumor in your body, then the quantum dot will be accumulated on that tumor. Now, if I irradiate infrared radiation on this rat, then here you see quantum dot is accumulated here. That means from here light will be emitted, and this light will be detected by an imaging machine, and we can tell that there is some tumor here. There's some tumor here. So the quantum dots are very much specific. You see, green quantum dot fields moves towards the nucleus of a breast tumor cells. These are the quantum dots. These are the quantum dots. It is going to here, and it will be accumulated on that tumor only. And if you give the infrared radiation on a body, then only in that, from that portion light will be emitted, and you can image, you can detect this portion by some sort of imaging machine, and you can detect the position of the tumor. You see here, if you, this is a quantum dot, it is accumulated on a tumor. ultraviolet light is off. It is like that. If you on the ultraviolet light, some quantum for some quantum dots, you require the infrared light. If you make on the ultraviolet light, you will see these quantum dots are giving colors. And you can imagine that if you see the color, if you can see the color, if you if you can see the light, then you'll see the shape and size of that substance. We are not seeing the tumor, but instrument can see this. So it can see this, and it can tell us about the shape and size of this tumor, and it can find cancer signatures also. Say for example, this is the cancer cells, and these are the healthy cells. The quantum dots will accumulate on the cancer cell, and if you give infrared light or ultraviolet light, they will show that that the cancer cells is here. It has this quantum dots has a unique property. Not all quantum dots, but some quantum dots has unique property to be accumulated on the cancerous cells. Now, you see the treatment, the cancer treatment. You see the if you attach medicine on that quantum dot, then this quantum dot will enter into the you see this quantum dot will enter into the cancerous cell, and the cancerous cells will be destroyed. But normally, say for example, what is the treatment of cancer? We generally use chemotherapy. In chemotherapy, the medicine destroys the cancerous cell as well as the good cells also. But here, as because we have attached medicine on the quantum dot, and the quantum dot will be accumulated only on the cancerous cell, then the medicine will destroy only the cancerous cell. You see, only the cancerous cells, the green cells are destroyed, not the pink cells. They are very good. You see, these are the cancerous cells, and these are the quantum dots with your medicine. Then it will destroy only this thing. But here you see it destroys these cancerous cells as well as the good cells also. Now, this is how quantum dots are used. So, what is the main thing of quantum dots? The quantum dots are some metallic semiconductors. The metallic semiconductors, you can use the metallic semiconductors for treatment or for diagnosis. How? Because this some type of metallic semiconductor is very much specific. They will only accumulate on the cancerous cell, not in the. They will not accumulated. They will not be accumulated on the on the on the normal cells. For that, we can use that quantum dots as nano devices. Now, next one is nanopore. So what do you mean by pore? Pore means a small, very small hole. If the hole size is, say, for example, 1 to 10 nanometer, then it is called nanopore. And in this nanopore, your electrical current may be can flow. Electrical current can flow through this hole. So why this is required, why nanopore is required, and how it is used? Let's see. There are different types of nanopores. First, I'll tell you there are different types of nanopores. One is biological nanopore, one is solid state nanopore. Biological nanopores means the nanopore is the nanopore made by biological substances. And the solid state nanopore means the nanopore which is made by researchers, or which is made by human. But biologically, that means self-assembly. This nanopore is made by self-assembly. Now, more recently, hybrid nanopores. That means a combination of these two are also done. Hybrid nanopores are proposed to take advantage of the features of the biological and solid state nanopore. It has some advantage. Solid state nanopores have some advantage. The hybrid nanopores, that is, you can call it composite. The hybrid nanopore has advantages of both the biological and solid state nanopore. Now, alpha hemolysin. Alpha hemolysin, it is, you see, it is a mushroom-shaped protein. Alpha hemolysin, it is a protein. Its shape is like that. It can form pore. It is a toxin that is secreted by Staphylococcus aureus. It is one type of bacteria. This alpha hemolysin is found from one type of bacteria called Staphylococcus aureus. This has a unique quality to form a pore on a substrate. If you place this type of this protein on a substrate, it can form a pore. So you see, this mushroom-shaped heptameric protein normally form pores in the membranes. Membrane in the substance, you have membranes inside in your body. There are various types of membranes in our body. This mushroom-shaped heptameric protein normally forms pores in the membranes. You see the pore. This is the pore. This is the pore which is formed by alpha what is called alpha hemolysin protein. This pore is formed in a lipid bilayer. That means in a lipid membrane, when you place this alpha hemolysin, then it forms pores of nano size. Now, why these nanopores are required? You see here, why these nanopores are required. Remember that it is a biological assembly. That means it is done automatically by means of proteins, different types of proteins. Actually, these nanopores are used to sequence the DNA. I told you yesterday what is DNA sequencing. DNA has four bases: adenine, thiophene, guanine, and adenine, thiosin, guanine, and uracil or something. So they have a sequence in a DNA. And what is that sequence? How can you measure? You see, DNA here, you see, DNA can be sequenced by threading it through a microscopic pore in a membrane. You have taken a nanopore here. You have a nanopore. Say, for example, this is a nanopore, and this pore is here. So what is the steps? You see, one protein unzips the DNA helix. Unzips the DNA helix. You see, this DNA is a double-strand helical structure. DNA has a double-strand helical structure, you all know. One protein, what does it? It opens the helix. And the second protein creates a pore on the membrane and holds an adopted molecule. And a second protein creates a pore. Which is the second protein? That alpha hemolysin creates a pore here and makes this DNA protein, makes this DNA thread to enter through this pore. Like it is like threading. It is like threading in a needle. First one protein opens this DNA helix, and next another protein makes a pore here and makes this DNA thread to enter into the pore. Now, a flow of ions through the pore creates a current. Now, you can create a current through this pore. Why it is required? To detect the bases. How to detect the base? Say, for example, DNA has adenine. So when adenine enters to this pore, the current experience of obstacle. You are flowing current through this pore, and adenine is entering through this pore. Then the current faces an obstacle. So this obstacle can give a sign like this. Now, next is guanine. DNA has four type of bases. First adenine, then say for example it is guanine. When guanine enters into the pore, it faces different types of obstacle. The current faces different types of obstacle because adenine and guanine are different compounds. So when adenine goes through this current, it will have one type of obstacle. The current will have one type of obstacle. And when guanine enters, the current will experience another type of obstacle. And this, you see, this obstacle is like that. If uracil passes through this pore, then it will face another type of obstacle. So you can tell which type of base is passing through this pore by seeing this spectra. Which type of base is passing? Say first adenine is passing, then guanine is passing, then uracil is passing, then thymine is passing. So the sequence is adenine, guanine, uracil, thymine. And then again thymine is passing. So the sequence starts from that thymine. So by this way, you can sequence the basis of DNA by means of a nanopore. You see, the pore is like that, the pore is like that, and the DNA is going through this nanopore. And if you pass current with this, you will you will face the current will face the obstacles by the bases, and you'll see a spectrum. This is the nanopore. Next one is nanoshell. It is a nanoshell means it is a shell. You see, it is a shell. It has a shell and it has a core. Have you ever eaten shell and core ice cream of igloo?
जी सर कैसे
Mhm. It has a shell, and inside it, it has a core, just like a choco bar ice cream, but the shell is hard. So the nano shell is, say for example, this is a silicon sphere, and outside it, it is a it is a gold sphere. So the shell is of silicon, and the core is of silicon, and the shell is of gold. This is this is known as nanoshell. So it's a type of spherical nanoparticle consists of a dielectric core which is covered by a thin metallic shell. That means the uh, the inside the inside substance is a dielectric material. Dielectric material means it is like an insulator, but the the shell material is a very good conductor of electricity. This type of How can you prepare this type of thing? You see, this is a silicon oxide nanoparticle. Uh, this silicon oxide nanoparticle, these are some amines, and you attach amines with the silicon oxide nanoparticle. That in that amine, you 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 attach the gold atoms. This is the this is actual This is the actual mechanism of nanoshell synthesis. You see, there is a core, and this is the shells. So, what is the requirement of nanoshell? If you give light, say infrared light, this nanoshell absorbs heat from the light and it becomes heated. This nanoshell absorbs heat from this light and it becomes heated. So, so you see what it is doing. It is a cancerous cell. Say, for example, these are the cancerous cells and these are the good cells. The nanoshells will be accumulated on the cancerous cell. And if you give infrared light, if you treat infrared light on that cells, on that cells you see here, these nanocells becomes heated and it will burn all the cancerous cell, but this good cells remains unaffected. So what is the quality of a nanoshell? There are two qualities of nanoshell. One, it absorbs heat when you give infrared light. And second, it can be accumulated on the cancerous cell. Hence, cancerous cells are the special types of cells. They are not like that normal cell. They are special type of cells. So it can be accumulated on the cancerous cell. So if you inject nanoshells in anyone's body, the nanoshells will be accumulated on the cancerous cells only, not on other cells. And now, if you irradiate infrared light on the body, only the cells which have a nanoshell will absorb this infrared light and be heated, and that heat will destroy that heat will destroy the your cancerous cells. That heat will destroy the cancer cell. This is the advantage. This is the advantage of nanocell. Now, next one is dendrimer. Next one is dendrimer. What are the dendrimers? The dendrimers are tree-like polymers. You see here. This is a dendrimer. This is a polymer. You see, there is a root of the tree, and these are the branches of the tree. These are the branches of the tree. These are the tree-like polymers. And that has a size of about 15 nanometer. So these are the nanoparticles. Tree-like polymers branching out from a central core and subdividing into hierarchical branching units. So this is called dendrimers. Dendrimers, the name comes from Greek word dendron, also called arboreals. They are family of nano-sized, highly branched, three-dimensional molecules. You see, these are like tree. These are like trees. These are like trees. We have one example of dendrimer. You see, this is a dendrimer. Its name is polyamidoamine. Its name is polyamidoamine. This is the root of the tree, and you see this is the root, and you see this is the branching. These are the branching. These are the branching. These are the branching of trees. These are the branching of trees. There's a very beautiful, your polymer, and the size of this dendrimer molecule is about 15 nanometer. So it is a nanomaterial. So how it is used? You can attach your medicines with with this branch, with this branch, with this branch, and you can use it in multi-purpose use. Say they have applications as vehicles for delivery of genes and drugs. They have medical applications for MRI contrast enhancer. It is molecular carriers for chemical catalyst. These are the carriers actually. You see, you can attach various things with these amine molecules. You can attach various things with these amine molecules. Say, for example, you have attached something in this region. You can attach something in this region. You can attach some other things in this region. So a dendrimer can be used for various purposes. One dendrimer can be used for various purposes. You see here, this is a dendrimer, and you can add some therapeutic agents here. You can add some cancer detector here. You can add some cell death monitor here. You can add some diagnostic materials here. So in one molecule, you can add different types of things in one molecule, and you can use it. So, and it is very much a nanomaterial. So you can make a dendrimer, and you can detect with the dendrimer. After detecting, you can treat with that dendrimer. And the last one is nanotube. You know what is nanotube? That carbon nanotube. You can insert medicines inside the carbon nanotube. You see there is a big hole here, and you can insert the medicine inside carbon nanotube, and you can carry this carbon nanotube can carry the medicine to deliver in specific portions. So these are different types of your your nano devices, or nanomaterials, or bio nanomaterials, whatever you can say, or nanobiotechnology, that are used in our body. So do you have any question?